Hello everyone, welcome back to AS Biology with Dr. Demi. I am Dr. Demi, and in today's video I will be concluding chapter 10 of the AS Biology syllabus. That means I'll be focusing on antibiotics and antibiotic resistance. If you haven't watched the videos preceding this one, I suggest that you do so that you have some context into what we have discussed so far in terms of infectious diseases, and also understand how antibiotics and antibiotic resistance might play a role in the spread of infectious diseases. So I'll just get into it from here on. So what is an antibiotic and how does it work? Well, what antibiotics do is that they interfere with the normal functioning of a bacterial cell. Something I have to point out here is that typically antibiotics do not work on viruses, simply because viruses are acellular organisms, so they don't necessarily have any form of metabolic process that's going on that can be interrupted. Viruses are simply made up of genetic material, protein coat, perhaps a lipid coat, and some receptors and enzymes that enable them to infect a host and to multiply themselves. Bacteria, on the other hand, have metabolic processes that go on within their bodies. They have cell wall synthesis, they have enzyme action, they have DNA synthesis and replication. They also have protein synthesis. So what antibiotics try to do is they try to interrupt one of these activities, because by interrupting them, the bacteria is unable to reproduce itself and cause a big infection. So an antibiotic in that sense is a drug that kills or stops the growth of bacteria without harming the cells of the infected organism. So usually antibiotics can kill off bacteria, and some of them can be wide spectrum antibiotics, which means that they would kill any bacteria in the vicinity where they act. That can be a bit dangerous for some people, especially if it affects your gut. So you would find that when you go to the doctor, when you're given an antibiotic, sometimes they give you a probiotic as well, just to replace some of the healthy bacteria that might be killed off by the antibiotic. So just again to show you some of the places where antibiotics would act, I'm just going to use a black pen here to highlight, or rather use a red pen, sorry. So for cell wall synthesis inhibitors, the antibiotics that are typically used, they are penicillin, cephalosporin, and vancomycin. To inhibit transcription, which is what allows for protein synthesis, rifampicin is quite common. Protein synthesis inhibitors, you have chloramphenicol, erythromycin, tetracycline, and streptomycin. So basically just looking at this image, which by the way I got from the course book that we're using, which is the AS and A-level biology course book by Mary Jones, you would find that there are so many different antibiotics that react or act in different ways on a bacteria that they are trying to attack. So let's dig into penicillin a bit. Penicillin is one of the most common antibiotics out there. It was discovered by Alexander Fleming in 1928. And what happened with Fleming was that he was working with Staphylococcus cultures. Staphylococcus is a different bacteria. And what he found was when he left the plate out, there was a mold that grew on one of the plates. And the mold, where the mold grew, the Staphylococcus did not grow. And what this simply means is that the mold had prevented the growth of Staphylococcus, and the mold came to become penicillin. Thankfully, we now take it in the form of tablets and not as a mold, because I don't think anyone would want to ingest mold. Penicillin, its mode of action is to prevent the cross-links between the peptidoglycan polymers of a bacterial cell. So remember when we discussed bacterial structure, or prokaryotes rather, at the beginning of this AS syllabus in chapter 1, we said that they have cell walls that are made of peptidoglycan. What penicillin does is that it prevents those units of peptidoglycan from linking to each other, so that the bacteria is unable to fully form itself. And if you want to go into a bit more detail as to how that is formed, the bacteria secrete enzymes that are called autolysins. What autolysins do is that they make little holes in the cell wall of the bacteria. And the point of those holes is to sort of extend the wall, because when those holes are made, the bacteria would make cross links or more peptidoglycan polymers to link to each other so as to extend the cell wall. And this is how the bacteria is able to grow in size. What penicillin would do is that it will inhibit the enzymes that causes those cross links to form. So when the bacteria secretes autolysins, the autolysins will go in there and make the holes, hoping for crosslinks to form that would extend the cell wall of the bacteria. Unfortunately, the cell walls, the crosslinks do not form if penicillin is present, and that means the cell wall of the bacteria would be weaker, and when it's weaker, it means that the contents of the bacteria would spill out, or the cell would burst because of the pressure that is exerted on the cell wall. So the cell wall would burst, the contents leak out, and that means the bacteria would die, because it would then be engulfed by the phagocytes and it would be digested, and that's the end of it. Now, antibiotic resistance is a very important phenomenon in our society today, simply because we have discovered that as time has gone on, some bacteria have developed resistance to certain antibiotics. So, for example, penicillin is not effective against Mycobacterium tuberculosis, simply because this bacterium has a thick cell wall, and it also has enzymes that are able to break down penicillin. So you'd find that some bacteria are just naturally resistant by virtue of their physiology or the way that they are built. Some bacteria also have mechanisms that pump antibiotics out of their cytoplasm. So simply they're saying, well, you just take a hike and they kick out the antibiotic, which means that it is unable to bind to its site of action, and as a result of that cannot cause the change or the treatment that is required. This is an image I have put here on the slide just to show you what antibiotic resistance looks like in the lab. So in the lab, if you decide to culture bacteria on an agar plate, you would take a plate that's clear as this one. What that white structure there, or the transparent thing, is called agar. Agar has nutrients that help bacteria to grow. And you would streak your bacteria, or you spread them across the plate. And typically when bacteria grow, they have this cloudy formation that you can see here. Now, what you can then do to test an antibiotic is that you take discs. These are paper discs. You take these paper discs, you soak them in that particular antibiotic, and you place them on the plate. Now, if the antibiotic is effective, you will see that around the disc there is a clear zone. This clear zone simply means that the bacteria is unable to grow anywhere in the vicinity of the antibiotic. So you can see here that there are many clear zones, which means that this bacteria is not resistant to the antibiotic that was placed on the paper discs. On the other hand, when you have antibiotic resistance, you can place the paper discs with the antibiotic on the plates containing the bacteria, and what you will see is that the clear zones are not as big as they should be, or in some cases, just like this one over here, there are no clear zones at all, which means that the antibiotic is not effective against the bacteria. Unfortunately, there are very many incidents of antibiotic resistant bacteria, and that is why many of the illnesses that we have today are not simply being cured by traditional medicine anymore, or should I say conventional medicine anymore, and that is because of the way we have used antibiotics, and that's something we will get into later on in this lesson. So bacteria can become resistant. The big question is, how do they become resistant? I mean, besides the fact that they might be naturally so because they have physiological mechanisms that prevent them from being susceptible to antibiotics, they can also become resistant by acquiring a gene that codes for a resistant protein. So antibiotic resistance in bacteria can be a result of the bacteria just acquiring a gene from another bacteria. So let's say bacteria A has a gene that allows it to pump out antibiotics, which means they have no effect on it. It can multiply that gene and actually give it to the bacteria that is closest to it, and as a result, that bacteria then gets the gene and is able to resist the exact same antibiotic. So you can call it some sort of community work, because they sort of try to protect each other. So for example, you have soil and pathogenic bacteria, and they have enzymes called beta-lactamases, which are able to break down antibiotics or protect them from antibiotics. Antibiotic resistance can also be due to a gene sequence in the bacteria spontaneously changing. In genetics, we would call this a mutation. So when the genetic material or the genetic sequence of the bacteria changes, it can code for a different protein that the antibiotic is unable to bind to. So let us say, for example, I'm just going to draw some images here. Maybe that would help. Let's say, for example, we have a bacteria that looks like that. And on on the surface of this bacteria, the protein would usually code for spikes that look like this. And so we decide, okay, we work in the lab, we're going to get an antibiotic that is able to bind to these spikes. So I'm going to draw the antibiotics, let's see, in this red color over here, but I'm just going to put a shape here. So this is our antibiotic. You can see that it has a shape that is somewhat complementary. My drawing is abysmal most of the time, but it is somewhat complementary to the spike on the bacteria. So when it binds, it's able to act on the bacteria. But now let's say something happens within this bacteria and its sequence changes. And now instead of coding for spikes, it is coding for semicircles, or let's just say it's coding for a different type of receptor on the surface. It means that when this antibiotic that we have that functions against the bacteria normally comes here, it is unable to bind to the surface of the bacteria, which then means it is unable to act. So the genetic transformations that happen within bacteria are actually very important in understanding how they develop resistance. Antibiotic resistance, like we said, because bacteria can acquire resistance from other bacteria, that transmission of the antibiotic resistance gene can either be vertical or horizontal, and we're going to have a look at that on the next slide. Something that is important for you to know is that bacteria have a single loop of double-stranded DNA. So it's a loop that looks like that, and it's double-stranded DNA, so it's just wound around, so it's one loop. It's not two different loops, it's just a single loop. And so it means that if there is a mutation in the gene sequence, resistance can be developed immediately. And that means that any antibiotic that you develop 5 minutes after would not be able to work. Also, because bacteria multiply very quickly. I mean, if you put bacteria in a sugar solution and you give them all the nutrients that they need to grow, if you go back within 6 hours, the bacteria could have grown from 100 different cells, maybe 100 million cells, simply because they have lots of food available to them. So what this simply means is that if that one cell becomes resistant, as it starts to grow through binary fission, because bacteria just split into two in order to grow, it means that in a matter of hours, it would have made hundreds of millions of the exact same type of cells that are now resistant to the antibiotic that you were planning to use. So antibiotic resistance is a very important issue. Just like we said earlier, you can have transmission either vertically or you can have it horizontally. So vertical transmission is transmission of resistance through binary fission, which basically means through reproduction. So bacteria reproduce asexually. That means they don't need to mate with each other and they don't make sexual gametes in order to reproduce. They simply split into two, and that means that each bacterial cell will now have a copy of the mutated gene and will therefore have the antibiotic resistance. You can also have horizontal transmission, and that is where there is a plasmid transfer from one bacteria to another bacteria, and we call that process through which that happens conjugation. Also, the interesting thing about this is that antibiotic resistance through horizontal transmission can actually happen between bacteria of different species. So you might be dealing with a bacillus species, for example, but you get to a point where you realize that the antibiotic resistance in bacillus is now being seen in staphylococcus, it's being seen in streptomyces. And what that means is that these cells have probably met with each other through conjugation and have now transferred antibiotic resistant genes to one another, and so they become resistant against the exact same type of antibiotic. This is just an image from the textbook that we've been using. It's the course book, AS and A-Level Biology course book by Mary Jones and a couple of other authors. And this is just an image from there to show you how vertical and horizontal resistance would happen. So in vertical transmission, you would see that you have a resistant parent cell. You can see that. And what you have there is a replication of the DNA loop as well as a replication of the plasmid. Now, when cell division happens, which simply means that the bacterial cell splits into two, you then have two cells that now have the antibiotic resistance gene, if it's carried on the loop or if it's carried on the plasmid, they also have it. So this is interesting because, again, like I said earlier, bacteria replicate very quickly. They reproduce very quickly. That means from this one cell, you can have 1 million cells in a matter of 24 hours. And that means by then it would be too late for your antibiotic to function against them. In horizontal transmission, it's a very different story, or a bit of a different story. So in horizontal transmission, you have a cell that is resistant to antibiotics, and you can see there that that cell has its DNA loop and it also has the plasmid. And this is a non-resistant cell. This non-resistant cell has a DNA loop, but it doesn't have the plasmid that carries the antibiotic resistance. What the two cells would then do is that they form what we can call a pipeline. This is what we call conjugation, by the way. They form a sort of pipeline whereby this plasmid is then replicated and it is transferred into the new bacterial cell. So that's the non-resistant cell. And that means that if the antibiotic resistance gene is sitting on the plasmid, this new cell also now has the plasmid and is then able to become resistant to the same antibiotic. And this can happen between bacteria of different species. So it is very interesting to see that even amongst different species, or between species rather, you would find that these bacterial cells exchange DNA in order to help each other survive. Now, some interesting facts about antibiotic resistance is that we already misuse antibiotics rather. Now, when you go to the doctor and you get an antibiotic because you feel really ill, there's an instruction that is usually given to you, and that is to make sure you complete the course of antibiotics, even if you feel better. Now, many people, once they feel better after day 3, they say, oh, you know what, I don't need to use that medicine. I'll probably keep it for when I'm sick again so that I can use it. That is very terrible practice, and you should not be doing that. If you have been doing that, now is a good time to stop. This is because if you don't complete the course of antibiotics, you actually give the bacteria room to figure out how the antibiotic works. And because the body of the bacteria is smart, just like our bodies are also smart, they can develop resistance through a genetic mutation. And once the genetic mutation happens, it means that antibiotic becomes less effective even in other people, because remember, they keep transferring their DNA to each other. So the more we misuse antibiotics, the more bacteria will develop resistance. So I often tell people, when you take an antibiotic, make sure, even if you're feeling better, that you complete the course until the very last tablet. Wherever there is widespread use of antibiotics, you will find that there is also very quick spread of resistance. There's an author that I usually recommend to students. It's Robin Cook, and he writes about pathogens and how they infect, but he writes about them rather in a very beautiful fictional way that helps you to just relate with the story. So it can be very interesting. So if you would like to learn more about pathogens, bacteria, do get a Robin Cook novel. You will find it very helpful in just thinking about how these can play out in the real world. Bacteria can also gain multiple resistance and transfer their plasmids to other bacteria, just like I said. For example, vancomycin was an antibiotic that was used to treat Enterococcus faecalis. Just from the name Enterococcus, that means it's something that has to do with the gut, and faecalis obviously has to do with poop. And the bacteria developed resistance and transferred that to Staphylococcus aureus, and that led to the rise of Staphylococcus aureus being resistant to vancomycin. The author I was speaking about, Robin Cook, actually wrote a book. The very first book I read, which I can't remember the title, was about Staphylococcus aureus being resistant. It was a beautiful story that was set in a hospital, so you might want to check it out if you can find the title. Antibiotics are used very frequently in food-producing animals, such as cows. So remember, when we discussed tuberculosis, we spoke about how it is caused by Mycobacterium bovis and Mycobacterium tuberculosis. Now, to prevent prevent Mycobacterium bovis from infecting cows, many farmers would inject their cows with antibiotics. So by doing that already, they're increasing the chances of more resistance in Mycobacterium bovis, because it is an overuse of antibiotics and obviously creates more problems for us when we eat those foods. So some guidelines for use of antibiotics. This again is from the textbook, the course book by Mary Jones and a couple of other authors. So using antibiotics only when necessary, reducing the number of countries in which antibiotics is sold without a doctor's prescription. In some countries, you can simply go to a store and say, well, I want this antibiotic, and they would give it to you. That is something that needs to be addressed simply because it causes more misuse of antibiotics. People are not advised on how to use them or when to stop using them or how to complete the course, so they simply just use them as they wish. Also avoiding wide spectrum antibiotics. When they say wide spectrum antibiotics, it means antibiotics that are able to attack different species of bacteria. Why this is important is that if you use wide spectrum antibiotics a lot, it means that if that bacteria or any of those bacteria become resistant, they will be able to transfer the same to other bacteria that are affected by that antibiotic. Making sure that patients complete their course of medication is also important, and they don't keep unused medication for themselves in future. Again, like I said, when you're using antibiotics, you don't say, oh, you know what, I'm feeling better, so I'm just going to keep this little bit until the next time I feel ill again, and then I'll use it. By then it's not going to be effective, so you have to complete the course. And also just switching up antibiotics every time you want to prescribe for a certain condition, so that the bacteria don't know what's coming. You hit them with a surprise every single time. And avoiding antibiotics in farming is also very important. It is better to cure infections in farming, because if you try to use antibiotics too much, then the resistance continues to spread and it can create a crisis in the world. And that is the end of antibiotics and antibiotic resistance, and also the end of chapter 10. That means we only have one chapter left on this channel that I am going to cover. I'm not going to cover the A-level biology syllabus, unfortunately, but I do hope that the AS biology syllabus has been helpful so far. The last chapter is called immunity, and in that chapter we will learn how the body fights infections. Until then, have a good time.